right, one more thing to talk about before we start talking about the process of protein synthesis is what, kind of the geography. Where is this happening? Okay, so it's going to be, what we're going to be doing is we're going to be copying the coding information that's on the DNA that is part of a chromosome. Remember that chromosomes are DNA wrapped around histone proteins. And we're going to be copying a particular gene at a time. So there would be just one part of the DNA copied. Chromosomes have hundreds of genes on them, so we're just copying like a little piece. The other thing to know is that the nucleolus inside the nucleus has the ribosome subunits. The ribosomes are going to be part of this process, and where the process actually happens is on the rough ER. So what is going to happen is we have to copy the DNA, we're going to copy it to RNA, and we're going to also take the ribosomes, and we're going to take both of those things outside here on the endoplasmic reticulum membrane system, and the ribosomes are going to assemble there, and in a particular ribosome, what's going to be happening is the protein synthesis process. So basically you could say the protein synthesis is happening in the rough ER. This is why earlier in the semester we said the rough ER is responsible for making proteins, all right? So that's just kind of generally what's happening. What we have to do is we have to take, first though, we're going to copy whatever the gene is over to RNA. And the way it's going to be copied is basically like the replication process in DNA, except for remember that RNA does not have thymine. It has uracil instead. So when we copy RNA from DNA, we're going to have adenine is going to be copied to uracil, thymine is going to be copied to adenine, cytosine to guanine, and guanine to cytosine. So our RNA is going to be looking like the base pair, except for with the exception of uracil instead of thymine, all right? So let's say if I had a particular codon on DNA, if I had that codon and let's say it was GAC, my RNA is gonna then be CUG. If I had a codon that was ATT, my RNA is gonna be UAA. So what I have to do first, just to reiterate, is I have to copy my DNA sequence, the gene that I'm going to be translating into one protein. So one gene equals one protein, basically. And I'm going to be copying that, and I'm going to be copying it in the form of RNA. And then I'm going to use the RNA on the rough ER to help with the copying process.